Good morning, everybody. Welcome to another instance of our RNA special interest group. I thought I'd just start with a quick intro to the group because I feel like it's been a couple of months since we've been through that. Um, so this group is a community for anyone interested in RNA 3D structure and what goes into the unified goal of creating a prediction method that's able to predict RNA 3D structure. So that includes members of the community like the experimentalists solving RNA tertiary structures, as well as what you'll hear about today, which is more high throughput methods for secondary/tertiary structures, um, as well as the predictors themselves and the assessors. So it's quite a broad community and we try to curate talks from people from every part of that community. There is a Google group, which I will post the link in the chat below, where when you join, you'll get the email reminders for these talks. Um, and then most talks will be recorded. So you will also find a YouTube playlist with all the recordings. And I will also link our Google spreadsheet, which has both the schedule of upcoming seminars um, as well as past seminars and the link to the recording if there was one. Um, and then finally, that same spreadsheet will have upcoming gaps in our schedule. So, for example, I think beginning in May we have openings. So if anyone's interested, they can sign up there. All right. So I will send those links in the chat when the talk begins. For today, we're going to be hearing from Shijun He, which has been working on the Ribonanza talk that you heard about. I believe it was in October from Riju. So he actually started his RNA journey in a Kaggle competition that the DOS Lab put on during the pandemic to predict RNA degradation rates and performed extremely well in that competition. So designing a machine learning algorithm to predict degradation rates. Since then, he's become a Kaggle Grandmaster. So he's at the top echelon of machine learning predictors in the Kaggle universe. He's also a PhD student in Texas A&M and has been helping us on the creating side for this Eterna Kaggle dual crowdsourcing effort for Ribonanza. So Shijun, go ahead and take it away.
All right. Thanks, Rachel, for the introduction. So today I'll talk about Right Bonanza, deep learning of online structure through dual crowdsourcing. Uh, wait, okay. So I guess this is kind of redundant. So Rachel has already mentioned how I got into RNA. It's basically through a cattle competition hosted about 3 years ago called Open Vaccine. I kind of did well and also co-authored a paper with the organizers and other competitors that did well in this competition. Yeah, so fast forward, I became a grandmaster, and also actually in my PhD, I started working on developing stable mRNA vaccines, and we actually got a lot of funding basically by using the ideas I got from Open Vaccine and some of the work from DAS Lab. And also actually also published my work in Open Vaccine as a separate paper, kind of detailing the design philosophy and some post competition results with the Open Vaccine data. And fast forward to about July last year, Riju reached out to me asking if I wanted to actually be a host for another competition rather than a competitor. And the idea is kind of to scale up with a lot more data than Open Vaccine, which could potentially help us achieve the dream of going from RNA sequence to RNA 3D structure. And obviously, I was pretty on board with it. I'm pretty happy that I didn't try to compete because it was a lot of fun being a competition host rather than a competitor. Um, and I want to quickly talk about how we're going to scale up to get a lot of data. Obviously, for RNA, we only have about 1,000 atomic resolution 3D structures. And doing cryo-EM is kind of too slow to get to the scale we need. And a promising candidate is actually chemical mapping, um, where chemical modifiers are used to kind of attack the mRNA molecules, and essentially the unpaired positions uh, will be able to be measured, and then we can kind of look at the reactivity measurements at each nucleotide position. So this gives us an idea of which nucleotides are paired and which are not. And nucleotides with high reactivity typically are unpaired, and nucleotides with low reactivity are typically paired. And this chemical mapping data is actually a one-dimensional data because we only get a signal at each nucleotide. But in order to understand RNA structure, we kind of want to know for each RNA nucleotide with which position the RNA nucleotides are pairing. And there was this hack called mutate map, where you iteratively mutate each RNA nucleotide to its corresponding complement, and you can see on the diagonal when you mutate some of these nucleotides, it has increased reactivity, indicating that upon mutation, that position becomes unpaired. And very interestingly, if you look at off-diagonal positions, sometimes when you mutate a nucleotide, you get a response at a different position with increased reactivity. And this usually indicates the corresponding position is actually pairing with the mutated position. So this is kind of a good way to help us map how the cellular structure of any given mRNA molecule. And you can see encircled are these sort of diagonal features that are indicative of cellular structure. So this actually has been extensively used to kind of map out cellular structure of RNA. And it'll be really nice if we can have a model that can basically perfectly predict chemical reactivity. And then, for any given RNA sequence, we can actually just do mutate map in silico to understand what kind of structure it's forming. And for this to happen, we need first we need data. So we want diverse sequences. Sorry, and we also want people to actually train machine learning models using these diverse sequences. And the way to do that is via dual crowdsourcing. So Eterna, basically Eterna and Kaggle. Eterna is this platform where people are challenged to design RNAs with interesting structures, and it's basically kind of like a video game. And Kaggle has been kind of a staple website for comparative machine learning. So by combining these, we can kind of, have the data and also have the models trained by probably some of the best, machine learning people from all over the world. Actually, this has been done before in Open Vaccine, where we use basically the same setup to predict RNA degradation rates. And fast forward to when the competition actually starts, we have about 1.1 million RNA sequences with 2 profiles at each position. And an important sort of design choice is that we have sequences that are created, that are, measured during the competition, which are also larger molecules in the training set. So this serves as kind of like a rigorous way to do testing. And you also, by having longer molecules in the test set, we can also test the generalization abilities of machine learning models. And from a machine learning standpoint, the task is to predict 2A3 and DMS reactivity and each nucleotide. So this is kind of like a classic machine learning sequence to sequence task, kind of similar in spirit to actually how a BERT was pre-trained. And we decided to use mean absolute error as a metric to assess the models. So first, I want to show kind of like the power of Kaggle community. Within a few days, this Kaggler called iFrost, and his real name is Maxime. He put up a notebook that actually scores pretty well using just a simple transformer. So this is actually really good because uh, it is a pretty strong baseline, but it's not strong enough where it begins to completely dominate the sort of like what kind of models people are using. Because in many cases, if people put up baselines that are too strong, uh, everybody would just end up using the same models, and that's actually kind of bad for the diversity of top models. So yeah, I also kind of briefly talk about the transformer. So back in the day, it's actually not so obvious. It was not so obvious that you want to use the transformer to model RNA. Actually, in Open Vaccine, I remember the sort of like popular baseline model that people posted publicly was actually an LSTM model. So it's sort of like a different paradigm. But nowadays, transformer is pretty much ubiquitous. It's used in everything, in AlphaFold, ChatGPT. Uh, all these models use some variant of the transformer. And how it really works is I always kind of thought of the transformer as learning um, a graph structure on a sequence. And the way to do that is by basically looking at all the pairwise interactions between the different things in the sequence. In the case of RNA, these would be the different nucleotides in the sequence. And, in the case of natural language processing, these would be words. Um, so one of the kind of notorious things with transformers is that sometimes it kind of has a hard time generalizing to longer sequences, in particular like sequences that are longer than what's available in the training set. In fact, some of the popular transformer models, such as BERT, cannot make predictions for sequences longer than 512 because they use absolute learnable position encoding, which basically means that if you have a sequence longer than 512, basically you just get sequence, you get items that are longer than the sequence limit, which basically becomes undefined. And we kind of put long sequences in the testing set because we wanted to test the generalization ability of models. And early on, we actually started looking at some of these predictions, and we saw an M2 prediction of one of the top early top competitors, which what was quite striking was that the model just really wasn't responding to any of the mutations uh, in any of the off diagonal positions. So this was kind of worrisome. And so like my hypothesis for why this happened was probably the comparator used some sort of absolute positional encoding. Um, so when the model saw a really long sequence and uh, it sees basically unseen positions, it would just not know what to do. So we ended up making a post specifically to kind of give the comparators an idea on how their models should do on longer sequences. In particular, this is a set of mutant map sequences with which you're supposed to be able to map out the secondary structure of tetraamine ribozyme. And it turned out that if you use kind of like my model from Open Vaccine, you get pretty reasonable uh, profiles from these. So we decided to kind of post these and to give the comparators sort of a way to do sanity checks on these longer sequences. And this was kind of really necessary because the competitors didn't really have any way to test their models on long sequences during the competition because during the competition we didn't have access to any long sequence data. So, and during the competition, we actually started to do various experiments with sort of our in-house model, which is called the RNA deformer. And it's sort of a non-vanilla transformer. And the key variation is that it uses sort of a pairwise representation that originates from base pairing probability predictions, in this case, Eternfold. And it uses 2D convolutions to kind of update the base pairing probability matrix or the pairwise representation matrix, and then add that as an attention bias into the multi-headed self-attention. So this is kind of a way to enhance the self-attention mechanism with basically like a really strong prediction, really strong secondary structure prediction. And one thing we were really interested in finding out was if we have enough data to kind of do away with these secondary structure predictions from Eternfold. So we decided to create kind of a variant of the RNA deformer where instead of inputting base pairing probability matrix at the beginning, we kind of just take that out and we only input sort of a 2D distance matrix that acts as positional encoding for the transformer. And as expected, when you don't have a lot of data, the sequence only model kind of just doesn't do too well. And as we use more training sequences, the model performance difference between the sequence only model and the model with BPP as input kind of narrows. And at full training data, the sequence only model gets pretty close to the base pairing probability input model. And one of the interesting things we found out was that there's probably some invariance when you flip the RNA sequence because I tried this augmentation method called flip augmentation, where during training, you flip both the RNA sequence and the labels, and you do forward and backward propagation. And during inference, you kind of make prediction 2 times, once with the sort of forward sequence, and again with the flip sequence. And for the flip sequence output, you kind of flip the output of that as well as a way. So it's kind of a way to do test time augmentation. And that kind of seems to help a lot, specifically for the sequence only model. All right. So next we try to look at how much data we need to get good M2 predictions. So as same as before, we tested 2 variants of RNA deformer. One using the base pairing probability matrix as input, one with only sequence as the input. So interestingly, the one with base pairing probability matrix as input seems to already do okay with M2, even with just 1,400 sequences. But it kind of gets a little better as we increase how many sequences we're using for training. On the other hand, the sequence only model actually cannot do M2 at all at 1.4 K sequences. As you can see in the bottom left, there's pretty much no response in any of the off-diagonal positions. However, when we moved on to 14K sequences, we began to see these diagonal features that are indicative of secondary structure. And when we increase the training data even further, we saw that the model is kind of getting even better at making M2 predictions, in particular, becoming these off-diagonal patterns become more well-defined as we get to 140K sequences. So this kind of shows that we're already at a scale with which a model that only takes the RNA sequence as input can do a fairly good job at mutate map in silico. Right. And then we move on to kind of looking at post competition, what kind of models really work well. And it turned out the models that work well are all non-vanilla transformers. And they're all kind of variants of transformer, but all the self attention mechanisms in these transformers are enhanced by some sort of pairwise representation. So you can see in the case of our deformer and the first place model, both of them use the Bayes-Meyer probability matrix as an input and create pairwise representation, and then use some sort of 2D convolutional module to update the pairwise representation. And very, very nicely, we actually saw another model in the top ranks that are very, very different from these models that use Bayes-Meyer probability matrices as inputs, which is essentially like an AlphaFold style model. In particular, the Evoformer, which the comparator that designed this model called the Twin Tower architecture. So the main difference between this model and the other top models is that this model only takes the RNA sequence as input and instead sort of creates a pairwise representation by combining the sequence representation with relative position encoding. So I also kind of want to kind of explain in a very technical fashion how these two methods differ. So the first camp of the models is basically use 2D convolution on the Bayes-Meyer probability matrix. And the key idea is that I think the Bayes-Meyer probability matrix can be seen as a sort of soft graph structure where it tells you which RNA nucleotides are actually paired in a probabilistic way. So this seems very, very much like a parallel to how self-attention works. Because self-attention also learns kind of like a probabilistic attention weight. So in open vaccine, I actually discovered a way to use the Bayes-Meyer probability matrix, which is first doing 2D convolution and then adding that to the self-attention function. This method seems to have some limitations. The first of which is that it relies on so conventional assembly structure prediction algorithms. In this case, the Turner-Fold, and it's quadratic complexity in space and time because it's kind of like operating on a 2D image. On the other hand, we have kind of the triangular multiplicative update, which is from AlphaFold. And the key idea for this module is actually to update each connection in the pairwise representation with all the other ingoing and outgoing edges to other nodes in the graph. So the key difference is that this representation is learned completely from scratch. But there are some downsides. The biggest downside is that this module is actually qubit complexity in time, which means it just becomes really slow as the sequence becomes longer. But kind of nicely, it's actually only quadratic in space, which means you'll kind of fit on the GPU uh, for probably up to a few thousand nucleotides. And there's kind of another sort of triangular module called triangular attention, which was also used in AlphaFold. Um, but no one really explored that, probably mostly because that has cubic, both cubic time and cubic space complexity. So it's really probably just too difficult from a compute standpoint to even experiment with that. And that kind of just turned out to be, I think, the biggest bottleneck for people. You know, the third place competitor who used the Twin Tower architecture with triangular updates kind of mentioned that using the triangular attention was just too expensive. So he kind of ended up not trying that. And I also want to kind of show how the model behaves. Uh, how I guess like the models that use Bayes-Coutts probability matrix as a feature differ from the model that only uses the sequence as the input. So these are basically Dirac-Fourier variants and their predictions on the sequence that forms a Kissing loop circled in red. And by doing mutant mapping in silico, you can actually see the sequence only model is reacting very nicely to the mutations in the solenoided positions, and not only in the upper triangle, but also in the lower triangle of the M2 matrix. Whereas the BEP model actually pretty much completely ignores the solenoid and just doesn't really respond to this kissing loop. And this is kind of generally the case when we looked at a lot of other structures that are also supposed to be solenoided. The models that use the Bayes-Coutts probability matrix are just pretty conservative on predicting solenoids, mostly because Eternal really doesn't predict solenoids. It only predicts nested structures. And so I kind of really felt like using BEP as a feature is perhaps more of a crutch, and it just leads to kind of like some limitations for the model. Although using BEP is actually a really good way to get pretty low mean absolute error. In fact, if you have two transform models, if you just add the Bayes-Coutts probability matrix into self-linear function, you kind of automatically score much better on MAE. All right, and one important question is, did models learn RNA structure? So we looked at the sequence, which is Moyers coronavirus frame shift simulation element. And when we look at main two predictions in RNA diphomer, it shows some pretty well-defined off-diagonal features that are indicative of secondary structure. And post-competition, we looked at how the top models are doing on this sequence. And it turns out that the top models pretty much converge in terms of what they think the secondary structure is for this particular sequence. And when we looked at the experimental measurements, it also agrees with what the top models kind of think about the secondary structure of this particular sequence. So this is nice. This means models are probably learning secondary structure, and it's responding nicely to these mutated positions. But on the other hand, there are still some sequences where top models are not completely convergent. In particular, this is actually a really long sequence. It's about in the 450 nucleotides um, with the 5' and 3' regions combined. Um, so, uh, sorry, because we know we have the 3D crystal structure of this sequence in the protein data bank, we can actually directly look at like the reference secondary structure right here. And when we look at the predictions for RNA deformer, we do see that it's predicting a lot of the secondary structure correctly, even sometimes some of the pseudonauts. Um, but it's also, predicting some wrong secondary structure. For instance, on the top right, you can see a wrong stem prediction right here. Um, and we also looked at top three predictions for this sequence. And we saw that sometimes these models are making actually the same mistake. Uh, but kind of nicely, the second place model predicts this tetraloop connection right here. Um, whereas the other models don't really do that. So this kind of shows that although top models certainly have learned secondary structure, um, they haven't converged. And so on some of these, I would say more difficult to predict sequences, top models are kind of just a little more uncertain than some of the easier sequences. And I kind of also wanted to quickly talk about sort of like selecting the metric, um, because that as kind of as a cow comparator, I really feel like a lot of times, really not a lot of thought was given to the metric selection process, and sometimes there would just be crazy shakeup, and which basically means that the results between the validation, your local validation, and public and private data are just completely seemingly random. So for us to select the right metric, we kind of decided to do like a very quantitative simulation where we looked at data from Open Vaccine and we tried a bunch of different metrics that all seem reasonable for this task. And then we measured the Spearman R ranking coefficient between the private scores and the public scores. So if this is really so, how cattle competitions work is that um, during the competition you have a public leaderboard, which is more or less like a validation set, and you also have a private leaderboard, where the results on the private leaderboard aren't shown until after the competition. So when people say there's a lot of shakeup, that kind of just means that the private leaderboard and the public leaderboard are very inconsistent, i.e., a low streaming line. So we really try to find a metric that would maximize stream R between the private scores and the public scores. And the open vaccine, after trying a bunch of metrics, realized mean absolute error was the best. And this also intuitive makes sense because mean absolute error is only dependent on the distance between predictions and labels, and is also like the simplest metric. And very nicely, after the Ravalanza competition, we did the same analysis with the top submissions Ravalanza. We actually saw that again, mean absolute error came out on top in terms of the Spearman R ranking coefficient. So this kind of just show that kind of whatever simulation we did kind of worked. So the comparators I think comparators had a good time competing because they can give reliable feedback from the public leaderboard. And another, probably the most important thing after the competition is for us to find a way to distill all the insights learned from the competition into a single model that is also very easy to use for downstream tasks. And we really want to kind of combine the insights from Rapidanza, not just from a modeling standpoint, but also from a data standpoint, uh, by kind of using some of the predictions from these top models. Um, and we also want the model obviously to be able to make 2D predictions. And this is actually something a vanilla transformer really struggles with because it only has a sequence representation. Uh, so it just kind of doesn't do too well with 2D predictions. And a lot of RNA tasks actually require you to make 2D predictions. And this includes, predicting secondary structure and predicting distograms, anograms for some of the 3D structure prediction algorithms, such as TR-Rosetta RNA. Um, and we also want to have just one model um, because typically in machine learning competitions, top comparators would ensemble a bunch of different models to get the most performance. But for downstream usage, that's probably just very inconvenient. You don't want to just post 10 different models. And it's probably just kind of hard to use if you do that. And it should be easy to use. So it should kind of just work. It shouldn't be complicated to kind of fine tune the model for downstream tasks. So we created what we call Rapidanza Net, which combines sort of some of the neural network design ideas from the top comparators. In particular, because we thought having the Bayes-Ring probability matrix as input is kind of a limitation. Uh, we kind of took inspiration from one of the top models that didn't use Bayes-Ring probability as input, and instead created a pairwise representation from scratch, basically using triangular updates, which is borrowed from the AlphaFold model. Um, Interesting, in this model, we still found 1D convolution to be necessary. Um, so, which is basically because AlphaFold does not use any convolutions. Um, but here we still found it necessary. And the nice thing about having a full-blown pairwise representation is that it's very easy to make 2D predictions because you can just add a linear layer on top of the final output pairwise representation and predict things like secondary structure and distograms. We also kind of decided to utilize the top models and their predictive capacities to do semi-supervised learning with these top solutions. So most of the sequences in Rapidanza are actually pretty low signal to noise. And what actually ended up happening is that only about 20% of training sequences are high signal to noise and can be used for training. And the rest of the 80% of the sequences are actually just not really usable for training. However, if we take the top models, and we take their predictions for these sequences that have noisy data, we can actually get a lot of data that are approaching experimental accuracy. And with that idea, we kind of got together about 1.9 million sequences as through labels. And we did self-distillation training. Distillation training with the 1.9 million sequences. And we kind of did it in sort of a slightly convoluted way, where we first trained a Rapidanza net with true labels of noisy training sequences from the top 3 teams. And then we actually combined the noisy train semi-supervised Rapidanza net with the store labels of top 3 solutions to retrain even stronger model. And we ended up with performance that's actually even beyond the top solution, which involved using 28 models to ensemble. Right. So after getting kind of really good MAE on the reactivity data, we kind of wanted to find out if RabbleAnswerNet is also good for other downstream tasks. So the first one we tried is basically to predict which sequence will drop out in the chemical mapping experiments. So in RabbleAnswer, as I mentioned before, only about 20% of the data is usable because some of these sequences just didn't get good data. So if we can kind of figure out, beforehand which sequences are going to get good data, we can kind of just do experiments only on the sequences that are going to get data, and that can help us basically increase the yield of our experiments. So Hamish fine-tuned RabbleAnswerNet, and it turned out to kind of work really well. It's actually out of all the models that we tested, it's the best. And he caught this interesting case of two RNA structures, two RNA sequences that actually have the same structure, but very different number of reads. So I think this might be due to a lot of repeat nucleotides, but it shows that RabbleAnswerNet has learned information probably beyond just chemical mapping data. And another interesting fine-tuning task is if we can predict RNA degradation rates using data from Open Vaccine. So this is actually a very similar task to what we have in RabbleAnswer. It's just predicting some value at each nucleotide position. And you can see the actual submissions in the competition. And RabbleAnswer is down here in green. And I actually created this model. It's called semi-supervised RNA deformer. That's actually better than any of the submissions in Open Vaccine. But it's actually worse than RabbleAnswerNet, despite the fact that the semi-supervised RNA deformer actually uses a 50 model ensemble with 12 different sets of secondary structure features generated with six secondary structure packages at two different temperatures. So this really shows the power of RabbleAnswerNet even as a single model, because it's beating some crazy ensemble. Okay, so this is kind of another task that originated out of the Open Vaccine competition. So the idea is if you sum up the degradation rates over all the nucleotides, they should correlate well with half-life. So for this set of data, we actually have 3 protein coding mRNA sequences, and each group there are like a lot of variants of mRNA sequences that encode for that specific protein, and we kind of see the RabbleAnswerNet is comparable with RNA deformer, which was the previous best on this test, and this is kind of a really good sanity check because sometimes these transport models kind of fail when you scale up to really long sequences. And in this case, we have sequences actually that are way longer than we have in the testing set, which is so here we have up to 1,200 nucleotides, and the RabbleAnswerNet still does pretty well. So another really important task is, can we fine-tune RabbleAnswerNet to predict secondary structure? So previously, we have been looking at M2 predictions, which are basically a way to map out secondary structure by doing single mutations. But it's actually kind of cumbersome to make predictions for like 100 sequences, 100 single mutants for a particular RNA sequence. So it would be kind of good if we can directly predict secondary structure. And it turned out that we can easily do that by adding a linear layer on top of the pairwise representation and produce a base pair score with which we can actually see what the model thinks a secondary structure is. And we kind of also use this algorithm called Hungarian to kind of discretize the pairing probabilities into an actual scientific structure that doesn't violate any of the scientific structure rules. And additionally, we actually found a simple heuristic to help us estimate how good scientific structure prediction is, which we call the EF1 score, which is basically a linear regression model on the predicted probabilities of the base pairs. And we also developed another EF1 score called EF1 cross pair, which assesses how the model thinks it's doing on base pairings that actually cross each other, i.e. pseudo knots. So we see for this particular sequence, the model thinks it's doing pretty well on both counts. So this is good. And when we move on to something that's probably more difficult to predict, which is Tetrahymena ribozyme, we see that the model actually still predicts these round stems, which are consistent with what we see on the M2 predictions, which is good because that means after fine tuning, the model probably didn't overfit to the fine-tuning training set, but rather it's still telling us what it has learned from chemical mapping data. And in this case, we actually get a pretty high EF1 score of 0.80, but the cross-pair score is actually kind of low. So this kind of shows that the model is kind of uncertain about some of these predicted pseudo knots. All right, so next we moved on to compare ribanzanet with basically a bunch of other algorithms that predict scientific structure. And we do find that ribanzanet does better than pretty much everything else. So it kind of achieves state-of-the-art performance on scientific structure predictions. And very kind of surprisingly, it does massively better on the Cas15 RNA sequences, which we don't really know kind of why yet. All right. So in Cas15, one of the top deep learning model, TR Rosetta RNA, kind of claim that if you just input better scientific structure to their model as an input, it'll do better in 3D structure prediction. And we kind of tested that on one of the Cas15 sequences where ribanzanet scientific structure predictions score an F1.99, and SPA RNA, which TR Rosetta used originally, only scored 0.54. And when we look at the 3D structure prediction, we do see a pretty good gain in RMSD. So the ribanzanet scientific structure assisted TR Rosetta kind of approaches experimental accuracy, whereas the SPA RNA scientific structure is kind of just lagging behind a little bit. So this kind of shows that ribanzanet can potentially help 3D structure prediction algorithms that use scientific structure as inputs. And we really try to figure out if the ribanzanet understands 3D. So one of the tests we did was essentially adding nucleotides in between a kissing loop. Supposedly, when you add these GC pairings, it should break apart the kissing loop if you add enough bases. Unfortunately, if you look at these scientific structure predictions from left to right, is illustrations of the original sequence, which is insert 0 bases, and the sequence where we inserted 5 bases, and the sequence where we inserted 15 bases. While the global EF1 and Crossfire EF1 scores do kind of go down, indicating the model is becoming less and less confident on the prediction. Um, the Sonar prediction doesn't disappear. Uh, so this shows that the model may have some understanding of 3D, but another component of how the model works is probably pairwise motif recognition. It sees kind of these sonar motifs together, and it just thinks it's pairing. This is actually pretty. It's kind of a signature of these top models, which use a combination of 1D convolutions and transformers. So it's not surprising that will happen. But I think RabbitAnsNet still understands 3D to some extent. All right. One of the interesting downstream applications that that we might be able to do with RabbitAnsNet is actually genome scan to discover interesting sonar structures. And this actually goes back to the EF1 score. So originally, I kind of came up with the EF1 score because I wanted to genome scan. And the idea is just to scan the genome with RabbitAnsNet and then calculate this score called OpenNOT EF1, which is inspired by OpenNOT score, which was used to design some of the sonar sequences for RabbitAnsNet, which is basically an average of the global EF1 and the crosspair EF1. So this kind of tells us how accurate the secondary structure prediction is for that particular sequence. And then we can take all the secondary structure predictions and all the sequences and rank them by OpenNOT EF1 score and the number of pairing positions. So we kind of added this weighting scheme using number of pair positions because some of the top ranking sequences before we added the weighting were kind of just like two stems that cross each other, which kind of just aren't going to be that interesting. So we decided to just add a weighting to that, and this does kind of help. And when we look at some of the top sequences now, they are kind of just really well structured and also have high OpenNOT EF1 score. And I also want to show an example of one of the top ranking sequences. So this is actually the second place sequence from the 200 Yucatan window scanning run. It's just some sort of pretty long range sonar with a really long stem. And when I actually input this sequence and secondary structure into the TR Rosetta online server. And we do see that TR Rosetta actually models this structure as a true knot, where the single-stranded region right here actually goes through the loop right here. And this one actually scores 0.88 OpenNOT EF1 score. And also 160 out of 200 nucleotides are paired, which is good. That's kind of what we want. Okay, and finally, we have all the notebooks and data available on Kaggle. We have the fine-tuning notebook to get state-of-the-art online secondary structure prediction. And you can actually see this run took 550 seconds. So it's actually under 10 minutes to get state-of-the-art secondary structure prediction. Obviously, we have the inference notebook, and also RabbitAnsNet training code and data are both available on Kaggle and GitHub. All right. Lastly, I like to thank everybody at DAS Lab. Has been really fun to work on this, and also the top comparators from Kaggle for providing really good models and really good ideas, which are really instrumental in helping us design RabbitAnsNet. And also shout out to Eternal developers and Eternal players, who have been instrumental in designing interesting sequences for the competition. Okay, so that concludes my presentation. I'll be happy to take questions.
Okay, thanks, Shahid. Okay. For everyone in the audience, please raise your hand if you have a question. And we have some in the chat that I'll get you started on. So the first part of Jacob's question, I think, was answered. But the second part was whether you can delve into a little bit more detail on how you go from that pairwise representation into the 2D structure for.
Okay, yeah, good question. Sorry, let me get to the slide. So, hello, can you hear me?
We can still hear you.
Okay. Thought I had an internet cut off. Um, okay. So if you look at this picture, uh, you can see the model outputs a pairwise representation at the end. So if you just add a linear layer on top of that, the outputs a single value per connection, you can then fine tune the model with basically binary quasi-entropy loss on the ground truth secondary structures. Um, so because we have a full blown low-end pairwise representation, that makes the process really easy. Because some of the other work in the literature actually would process an RNA sequence with the transformer or STM first, and then do something called outer concatenation network and 2D convolutions to be able to produce a 2D prediction, which is kind of just really inconvenient and also doesn't work as well. Um, and in our case, because we have the pairwise representation, it's just very easy to make 2D predictions.
I have a future-looking question for you. So, there's obviously still points that can be improved upon. What do you see as the top ways that the community can make that improvement to, for example, more correctly predict complex RNA secondary structure like tetrahydrobiopterin? What needs to happen for that improvement to happen?
I think probably the most obvious thing is getting more data. In particular, I guess long sequence data. Um, in terms of modeling, um, I think probably just making the model bigger will help. Although with some of these modules that we're using, which have qubit complexities, it's actually pretty hard uh, to make the model bigger. Not only because of the model will need more compute just because it's bigger, but also because when you have longer sequence as training data, due to the qubit complexity scaling, it kind of just makes everything really slow. Uh, so yeah.
No, I have a question. Maybe I missed at the beginning, but so there is DMA's data and shape data, and are they combined at the very, very top, or did people try to use just DMS data or shape data just to see if the results are different, or actually combining two sources of the data actually they bring better performance?
Um, that's an interesting question. So I guess you're kind of talking about modeling them separately. So basically using something like using two models. Yeah. So actually nobody ever really tried that, partly because it would take twice the amount of time to do the experiments. So the predominant sort of formulation is to train a model that predicts both values simultaneously, which kind of makes sense because they're pretty correlated. And actually, like no one really tried having separate models, which kind of makes sense because it just takes way longer to do experiments.
Yeah, so how the data is combined as an input? from
So,
two different from
well,
two different sources?
uh, so they're used as labels for training, right? So
Okay.
it's basically like training a machine learning model that kind of does multitasking and predicts two values at each nucleotide instead of just one. And this was actually also done in the previous competition, Open Vaccine, where we actually had five uh, values at each position because there were degradation rate measurements at some various different conditions, such as higher pH and higher temperatures. So in that case, it was actually would have been even worse in Open Vaccine to try to have separate models. I actually remember trying that during the competition, um, and I think it was worse than having a model that just predicts, all the different values simultaneously.
Okay, I will read the question from Grzesiek because I think it was partially answered. Because how difficult would it be to include other types of training data available? For example, known secondary structures to the model.
Yeah, so this is actually something we kind of try to do during the competition. We actually got all the chemical mapping data compiled on IMDb, which is a database of chemical mapping data, which involved many other different types of chemical agents. Um, and it turned out, according to the comparators, it didn't end up being that useful. But if I were to use them, I would probably just make a model that predicts, let's say, 20 different chemical mapping reactivities at each position, um, and just do training that way. That seems like it would probably be better, but none of the comparators did that.
Okay, maybe I will read one more for Grzesiek, from Grzesiek. Does ReBonanza.net predicts oligomers, like dimers formation?
Uh, Maybe I don't know. This actually I don't know. So we have I guess we have the fine tuning notebook. So if you have any data, uh, you can try to do this.
Yeah, probably now it works for, yeah, just sequence,
Yeah.
not explicitly dimers. Yeah, thank you.
I think we've reached our time. So thank you again for this wonderful talk and kind of summarizing the results of Red Bonanza. Very exciting to hear. We'll meet again in 2 weeks, the 27th of February, um, to hear a talk by Navjot Tor, I believe it is. So we will see you guys all then. And feel free to email me and Magnus about any questions you have or any recommended speakers. All right.
Thank you. A very good talk.
Bye, everyone.
Thank you. Thank you for having me.